Hello, everybody, and welcome to chapter 23. In this chapter, we're going to talk a little bit more about the subject of evolution. We're going to talk about, more specifically, we're going to talk about the evolution of populations and what happens in populations when the prevalence of certain genes changes. And we're going to look at a lot of examples as well from different areas of the world and different populations and things. The other thing I want to mention just at the outset is that if you happen to have a fear of snakes, there will be a few pictures of snakes in 2 or 3 of these slides. And I mentioned that because I knew someone once who had a real severe fear of snakes. And even seeing a picture of a snake was scary to this individual. So just want to mention that there will be pictures of snakes here as well. And I don't know, if you've got a fear of rodents, there's also going to be pictures of mice. So, we got something for everybody here, I guess. So anyway, to jump right in, then talking about evolution and the smallest unit of evolution. What is the smallest unit of evolution? So they start off this chapter by emphasizing something that was one of the most important things that we covered in the last chapter, which was evolution and natural selection. Natural selection acts on individuals. But only populations actually evolve. Only the populations are what evolve, not the individuals. And that's because evolution is actually a change, a genetic change in a population from one generation to the next. So evolution is actually, individuals. In the natural world, individuals are not going to be changing their genes. That just doesn't happen in the natural world. But populations can actually change their genes from one generation to the next, and over the course of many generations. And most populations are changing genetically somehow over the course of several generations and more. So there's a common misconception that individuals survive. Individuals don't survive. Individuals are subject to natural selection, but populations are the only things that actually evolve. So really the smallest unit of evolution is actually the population. And a little bit later in the chapter, we'll define specifically what a population is. Even though you've probably got a pretty good intuitive idea, common sense idea of what a population is. But anyway, we'll define it formally then later on in the chapter. A good example then of evolution acting on populations is that a certain type of finch called medium ground finches in one of the islands in the Galapagos Islands during a drought, the only plants that really survived well were plants that produced large seeds. And so during a drought that was observed on Daphne Major in the Galapagos Islands, only the large-beaked birds were more likely to survive because mainly there was only large seeds that were there. And remember, the finches in the Galapagos Islands were one of the things that Darwin actually observed when he was making all his observations when he was on a voyage in that ship around South America, but pretty much going around the world actually. And here's a good picture of one of those large-beaked finches trying to crack a seed there. I'm assuming that's a seed. That could be an insect there. It's kind of small. I don't know, but we're just going to assume that's a seed that's trying to crack. And here's another, look at that large, very, very impressive large beak. Just look at that beak. And so, in 1976, the before the drought, the average beak depth or beak size was, a little bit less than 9 millimeters, but then it was almost 10 millimeters then after the drought. So they did some careful records of these birds. So the finch population. So again, to emphasize the point, the finch population evolved, not the individual members. The individual members suffered, competition and natural selection and things, but the population itself was what evolved. And here we're going to look at, now here we're going to define something else, microevolution. Microevolution, in other words, evolution on kind of a micro scale. Evolution is just a change in allele frequencies in a population over generations. And so now they don't go into macroevolution here, but if there's microevolution, then there's probably macroevolution, right? Well, there is macroevolution, but macroevolution is changing from one species to another species. But microevolution is not necessarily producing new species, but it's just the change in allele frequencies. And what we mean by allele frequencies is, how common a certain allele is in a population. If you've ever done anything, in math class, if you remember them talking about frequency distributions, frequency distribution is kind of like a bell curve graph. A frequency distribution is just a graph that shows how common something is. And I'm trying to, I don't think there's any, we don't have any frequency distribution. Oh yeah, here we go. So here's a frequency distribution. It means that there are a lot of individuals that are right here in the population, but not very many individuals that are, we're looking at, mice, and we're looking at, how dark the coat color is. Here's a very light coat color. Here's a very dark coat color. Here's a coat color that's kind of in the middle. This shows that this is a frequency distribution. It's showing how many individuals have these different coat colors. And so individuals with there's very few individuals with a very light coat color, and there's very few individuals in a population with a very dark coat color. But there's a large number of individuals that have kind of an intermediate coat color. So this is a good example of a frequency distribution then. And so how common this coat color is in this population of mice. So that's why, here at the beginning, they're talking about the fact that microevolution is the change in allele frequency in a population. Does an allele get more frequent or less frequent? In other words, more common or less common in a population over generations, from one generation at a time. And remember, an allele. An allele is one form of a gene. For example, in Mendel's peas, there was a gene for the color of the peas, and one allele caused the peas to be yellow, and another allele caused the peas to be green. Or in Mendel's peas, one allele caused the plant to be tall, and another allele of a certain gene caused the plant to be short. So that's what we mean by alleles. So a little review of of General Biology 1, the first semester of General Biology. So anyway, a change in the allele frequencies in population over time, that's microevolution. And, the thing is, most actually, the discussion question talked about creation versus evolution, but actually most creationists would concede that, most creationists do assume that, allele frequencies change in a population. And so, most creationists have no problem with microevolution, as it's defined here, anyway, changes in allele frequencies. because you can see that all the time. It's easily observable, and we'll look at that in a number of things here in the chapter. There are three main mechanisms that cause change in allele frequency from one generation to the next. Natural selection, which we've already talked about, and then something called genetic drift, which we'll get in just a minute, and then gene flow also. So these three things cause changes in allele frequency, but generally only natural selection will consistently cause adaptive evolution. In other words, evolution that improves the species. Remember now in chapter 22, we talked about the fact that variation within a species, variation within a population, more specifically, is what makes evolution possible. So variation in inheritable traits, traits that are controlled by genes, not just the environment. Variation in heritable traits that can be controlled by genes is necessary. It's a prerequisite then for evolution by natural selection. And Gregor Mendel's then, whose work on pea plants, provided evidence of discrete heritable units, genes. Gregor Mendel actually, provided evidence that there are heritable units or factors, he called them. He didn't actually coin the term gene. That was later on, but his work showed definitively that for the very first time that there is a definable, a discrete, in other words, definable and definable and definite heritable unit or factor that causes genetic change. So genetic variation. Genetic variation then among individuals in a population is caused by differences in genes or other DNA segments. And a phenotype, you might remember this from General Biology 1, but a phenotype then is the product of an inherited genotype and environmental influences. Remember, genotype is what alleles does an individual have. Phenotype is what is the product of those, what's being produced by those alleles. For example, in going back to Mendel's peas, if the genotype is two alleles for yellow seeds, then the phenotype would be yellow seeds. A good example then, or if the genotype indicated that the blossoms would be purple rather than white, then the phenotype would be a purple blossom. Okay, so natural selection then can only act on variation that has a genetic component. And so natural selection can only act if there's variation there as well, like we saw in the last chapter. So some phenotypic differences are determined by a single gene, just one gene. And they can be classified as either on an either or basis. In other words, again, going back to Mendel's peas, the pea color, either yellow or green, that's actually determined by just one gene, one single gene. And it's an either or state. It's either green or yellow. It's not anywhere in between. And so there's a lot of traits can be, classified as either or. You have either blue eyes or brown eyes or green eyes, or yellow, Okay, so other differences then are determined by the influence of two or more genes that vary along a continuum. for example, hair color. I mean, you could have hair that's, kind of blonde or sort of blonde or kind of brown or very brown or dark brown or light brown or somewhere in between blonde and red, so a lot of things vary along a continuum, or your height is controlled by genes, but, so you can be, but the height of people varies along a continuum as well. So a number of different things, the shape of your nose varies along a continuum. The shape of your ears varies along a continuum as well. So there's a few different types of inheritance, then a few different types of phenotypes. And so now here's a good example. Horses. The color of horses. That's a good example of. That's a good example, very good example of variation within a population. Genetic variation then can be measured as gene variability or nucleotide variability, either one. It can be variability in the genes, or it can be variability in nucleotide variability. In other words, the sequence of the nitrogenous bases in DNA or RNA. For gene variability, something called average heterozygosity. We won't really get into that too much in this chapter, but for gene variability, then average heterozygosity measures the average percent of loci that are heterozygous in a population. And when they say loci, loci is plural and locus is singular, and a locus is a specific point on a gene. Well, actually, a specific point on a chromosome, actually. And a lot of times we're talking about genes here. So nucleotide variability then is measured by comparing DNA sequences of two or more individuals and seeing how much they have in common and how much are different. And this nucleotide variation, this variation in nucleotide sequences, actually rarely results in phenotypic variation. And we'll see that a little bit of that later on. Most of the differences occur in non-coding regions. If you remember, in a gene there are sequences called introns that don't have the code for some sort of protein, some sort of usable protein, but then there are regions called exons that do carry the code for certain proteins or useful RNAs. So variations that occur in coding regions, in other words, the exons, actually rarely change the amino acid sequence of the encoded protein. And for one reason, more than one codon can actually code for the same amino acid, as you saw in General Biology 1. So here you've got, for example, here you've got an exon. So that has the code for a protein that's being coded for in this gene. And then here's an intron. And this intron will be spliced out before the exons are put together to be used for messenger RNA. And so here's in these two places, DNA has been inserted. And here at this point, along that intron, there's been a deletion of a certain number of base pairs. And here's been a substitution. So substituting one base for another base. For example, maybe an A has been substituted for a T, or an A has been substituted for a C or as a G. So that's what they mean by substitution. So, so again, variations that occur in coding regions rarely change the amino acid sequence, but it does happen. And here's what we just looked at. So some phenotypic variation does not result from genetic differences among individuals, but rather from environmental factors. For example, if a person has the genes for being really tall, but maybe they have a poor diet when they're small, when they're a kid, and so they don't really grow to be as tall as they could be. And their poor diet is an environmental influence. But so basically, only genetically determined variation can actually be worked on by evolution, because evolution is a change in the genetics of a population from one generation to the next. And so here's an example then of some caterpillars. Here's one form, and then here's another form of a caterpillar, and it's kind of camouflaged as a twig. So we should look at sources of genetic variation. So new genes and alleles can arise by either mutation, like we've seen before, or something called gene duplication. And gene duplication is kind of rare, but it's a case where a whole gene is actually duplicated, so that where you had one gene before, now you have two genes on that chromosome. And sexual reproduction can produce a lot of genetic variation by recombining existing alleles through, because you get half, basically, you get half of your alleles from your mother and half of your alleles from your father, and there's a new combination. But also, if you remember the phenomenon of crossing over, chromosomes can cross over during meiosis and exchange genetic information, and that causes new combinations of alleles as well. So sexual reproduction then can cause a lot of genetic variation, and so that's generally good for a population. Okay, here. Just for a second there, I lost my place. So new alleles then can be created by mutations, and mutations are basically a change in the nucleotide sequence of DNA. And but only mutations in the cells that produce gametes can be passed to the offspring. Because if a mutation happens in a skin cell, for example, well, that can't be passed on to the offspring because those genes are not going to be passed on. Only mutations in the cells that produce gametes, egg and sperm, can actually be passed on to the offspring and be a change in the genetic material for the next generation. A point mutation is a change in just one single nucleotide in a DNA sequence, and that's the most common type of mutation. And so now, point mutations then, point mutations that change the phenotype are often harmful. A lot of times they're harmful. In fact, they're more often harmful than helpful. And that's one of the weaknesses of the evolution argument. If evolution is arguing that, well, mutations cause variation in a population, and the natural selection works on those mutations to improve the population, well, that's all fine and good. But if there are more harmful mutations than beneficial mutations, well, how is evolution really able to, improve a population? Anyway, so harmful mutations, though, can be hidden from selection in recessive alleles, because for a recessive allele to actually affect the phenotype, you've got to have two of those alleles. You've got to be a homozygous for a recessive allele. So you can be a carrier for a recessive allele, but yet that recessive allele is not, causing any effect on the phenotype. So that's why they say harmful mutations can be hidden from selection if they're recessive. Mutations that cause a change in the phenotype are sometimes, though rarely, admittedly rarely, beneficial. So there are a lot more harmful mutations than there are beneficial mutations. But natural selection is sorting them out as much as it can. But the thing is, I'll let you in on a little secret, though. If something is just slightly harmful, natural selection has a hard time totally eliminating it from the population. Anyway, the effects of point mutations, then, remember, changing just one nucleotide, can vary. Point mutations in a non-coding region, that one that doesn't code for a protein or a useful RNA, are pretty much result in neutral variation. And there's really no selective advantage or disadvantage. And so those types of neutral variation point mutations usually pretty much hang around pretty much permanently on the chromosome, because they're not taken out by natural selection. Mutations to genes can be neutral because of the redundancy of the genetic code. In other words, there's more than one code for more than one codon that's code for any given amino acid. That's what they're referring to here as redundancy in the genetic code. So chromosomal mutations, mutation of a whole chromosome. Chromosome mutations that delete, disrupt, or rearrange a number of loci, especially if they're genes, are typically harmful, almost always harmful. Duplications of duplication then of small pieces of DNA increases the genome size and is usually less harmful. The genome, if you remember from the first semester of general biology, a genome is the full sum total of all of the genetic material that a species has. Duplicated genes can take on new functions by further mutation. If you duplicate one gene to two genes, and then one of those genes mutates to some other form, well, then those two genes can diverge in terms of what they're used for. For example, then, so an ancestral odor detecting gene has been duplicated many times. And so humans have 380 copies of the gene, but mice have 1,200 copies of the gene. And so they may be able to detect that odor better than humans. Mutation rates are low generally in animals and plants. The average is about one mutation every 100,000 genes per generation. But we have millions of genes, so there's more than 50 mutations generally in a species. There's more than 50 mutations from one generation to the next. Mutation rates are often lower in prokaryotes, but short generation times allow mutations to accumulate rapidly in the populations. And so what are prokaryotes? Do you remember what prokaryotes are? The prokaryotes are the bacteria and the archaea, domain bacteria and domain archaea. Viruses that are very primitive, even more primitive than bacteria, have both high mutation rates and short generation times generally when they're actually inside a host. So in species that reproduce sexually, most genetic variation then results from recombination of alleles, like we talked about a few minutes ago. Sexual reproduction can shuffle existing alleles into new combinations by crossing over, that I've already mentioned, independent assortment, and fertilization. And so, and you can watch the animation then. Watch the animation that talks about genetic variation from sexual recombination.
In sexually reproducing organisms, three processes lead to most genetic variation: independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of one chromosome inherited from the father and one from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are two different ways that each chromosome pair can line up.
You see that here.
In the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being, with 46 chromosomes, more than 8 million combinations are possible.
So just by the process of meiosis, then 8 million combinations can be in just one individual can be formed. So watch the rest of the video then, and they'll talk more about crossing over, independent assortment, and fertilization. Genetic variation then. Now we're going to talk about Hardy-Weinberg equation. We're going to talk about populations, evolution then, and how things work in populations. So genetic variation then is required for a population to evolve, but it doesn't guarantee that it will. Just the fact that there's genetic variation does not guarantee that evolution will happen. So there are other factors that cause evolution have to be at work for a population to evolve. And let's look at that. Population. So we're defining a population then. It's a localized group of individuals of the same species that are capable of interbreeding and producing fertile offspring. And of course, if they're of the same species, they are, capable of interbreeding. Populations are not always geographically isolated from other populations of the same species, but individuals typically only breed with members of their own population. So there could be a number of separate populations of the same species. For example, mountain lions are found in certain areas here and there, or bighorn sheep. Bighorn sheep are found in certain areas here or there throughout the mountains. But they are geographically isolated, though. And so here, for example, you see a herd then up in Alaska, up in an area that borders Alaska and Canada. You see there's two different herds then of caribou then. Here's the porcupine herd in the porcupine area. And then there's the 40-mile herd in the 40-mile area then. And up close view of the porcupine herd, up close view of the 40-mile herd of caribou. So a gene pool then is all of the alleles for all loci in a population. And a locus is fixed. It's called fixed then if all the individuals in a population are homozygous for the same allele. A locus is fixed if there's only one allele at that locus, basically. So if there are two or more alleles for a locus, then if the species is diploid, then diploid individuals can be either homozygous or heterozygous. Diploid means that you've got two copies of each gene, and you've got two copies of each gene because you've got each chromosome comes in a pair, and for most eukaryotes, that's the case. Most eukaryotes are diploid. So most plants, animals, fungi, protists, things like that are going to be diploid then. So if a gene has two or more alleles, then diploid individuals can be either homozygous, have two alleles that are the same, or they could be heterozygous, have two alleles that are different. And here's a good example then for the flowers, like the snapdragon flowers. This has there's a gene for the color of the flower, and this individual is homozygous for the allele that causes red flowers, and that's why they have the R here. So basically that gene then on one chromosome has the allele for red flower, and that gene on the chromosome in the other chromosome in the chromosome pair also has the allele for red flowers. And then this individual is also homozygous because it has two alleles that call for white flowers. And then this individual is a heterozygote because it has one allele for red flower and another allele for white flower. And this is incomplete dominance. And so both red pigment and white pigment are produced, and so that produces an overall pink flower then. So that's what they mean by then diploid individuals then being either homozygous or heterozygous for any one gene. So the frequency of an allele, in other words, how common an allele is in the population, can be calculated in a number of ways. So when you're calculating frequency of an allele, one thing you have to remember is that for diploid organisms then, for diploid species, the total number of alleles at a locus is the total number of individuals times 2. Because there's 2 different alleles then at each locus. The total number of dominant alleles at a locus is 2 alleles for each homozygous dominant individual plus 1 allele for each heterozygous individual. And then the same logic applies to recessive alleles. You'll see this when we get into some equations. Okay, so by convention then, if there are 2 alleles at a locus, then P and Q are used to represent the frequencies. And the frequencies of all alleles in a population will add up to 1. So basically, the total frequency of allele P plus the total frequency of allele Q is 1. It's kind of like, when you have a certain percentage, when a population is broken down by percentages, all those percentages have to add up to 100. In this case, these are going to be proportions, and so all proportions have to add up into 1. And so here's an example. Okay, let's say that there are 320, and we're looking at the wildflowers. They're probably the snapdragons, but that back here in this example that we just looked at, they are incompletely dominant for flower color. And let's say, okay, in a population there are 320 red flowers, 160 pink flowers, and 20 white flowers. Okay, now so the red flowers then have 2 R's, 2 R alleles. The white flowers have 2 W alleles, and then there are heterozygotes. Okay, so basically, then these individuals each have 2 white alleles. So if you're looking for the total number of red alleles, you can't just look at the red flowers because every individual that's heterozygous for flower color has 1 red allele then. So now if you calculate the number of copies of each allele in the population, then there's 320 red flowers, but each of them has 2 red alleles, okay? So basically then you get, so you have to multiply the number of red flowers by 2 since each one of them has 2 red alleles. But if you're also looking for red alleles, you also have to add in the total number of those that are heterozygotes, because each heterozygote has 1 red allele, okay? So the way to find the number of copies of the red allele in the population then is to take number of red flowers multiplied by 2, since each one of them has 2 alleles, and then multiplied by 160, the number of those that are heterozygotes and have 1 copy of the red allele. So basically you get a total of 800. And then you do the same thing for those looking for the total number of white alleles. Take the total number of homozygotes that have 2 white alleles, and you divide that by 2, or you multiply that by 2. And then, but all of the heterozygotes, remember, have 1 white allele as well. So you add in the number of heterozygotes. And in this example, it comes out to 200 then. So what they're saying here is basically, this is what we have on this slide basically is a practical example of what they were talking about on slide 32 when they said that the total number of dominant alleles at a locus is 2 alleles for each homozygous dominant individual plus 1 allele for each heterozygous individual. And then the same is true for recessive alleles. Of course, in this example, it's not dominant or recessive because it's incompletely dominant, but still, this is a good example because it shows you how to easily calculate the numbers of copies of each allele in that population or in that sample that you took of the population. And now you should calculate the frequency. In other words, and remember what frequency is. The word frequency, what does the term frequency mean? Term frequency just means how common is that allele in the population, or how often does that allele occur in the population. So to calculate the frequency of each allele, or how common each allele is, basically, so remember we have P and Q in the original, in the original equation here, slide 33, we've got P and Q. And so, P is going to be, we just chose P to represent the red allele, and then Q to represent the white allele. So the P basically then, is the frequency of the red allele, and that's 800. And so we want to know the proportion. You could think of it as a percent, but we want to know the proportion of the population that has the red allele. In order to do that, we take the number of those in the population that have that red allele and divide it by the total number in the population. So we're basically getting a percent, but actually this isn't percent, it's proportion here. And so basically the proportion of the population that has the red allele, well, the proportion of the alleles of this gene in the population is 0.8, and the percentage of it is 80%. And then remember when you're doing this, well, okay, so for the white allele, it's actually the proportion of the population is 0.2 and 20%. So all 20% of the alleles at that gene are the white allele. Remember, the sum of all the alleles is always 1, and in percentages it would be 100%, of course. And so proportion of 0.8 and a proportion of 0.2 added together equals 1. That's kind of a good way to check your work, because if you add the two proportions together and you don't get 1, then you've done something wrong. So now we're going to talk about what's called the Hardy-Weinberg equation. And it's one of the most important concepts in population ecology and also population genetics. Hardy-Weinberg equation then describes the genetic makeup that we expect for a population that is not evolving at a particular locus. So it describes what is going on, what the state of affairs is going on if a population is not evolving, in other words, not changing at a particular locus. Remember this word locus. So, loci, locus is singular and loci is plural because it's Latin. So it's kind of like alumnus and alumni. So if the observed genetic makeup then of a population differs from the expectations under Hardy-Weinberg, then that population may be evolving. Because remember, Hardy-Weinberg describes the conditions when a population is not evolving. But let's look at some examples then here. In a population where gametes contribute to the next generation randomly, and Mendelian inheritance occurs, along a simple Punnett square, allele and genotype frequencies remain constant from generation to generation. And such a population is in Hardy-Weinberg equilibrium. But we'll look at an example. We'll look at an example, and we'll see more clearly what they're talking about here. Hardy-Weinberg equilibrium then describes the constant frequency of alleles, a situation where the frequency of alleles is not changing in a population, or we think of it as a gene pool. So as an example then, what if you have a population of 500 wildflowers? And so the population of 500 wildflowers, then they would represent a total of 1,000 alleles, if we're looking at their red and white flower color alleles again. And let's say the frequency of the red allele is 0.8, and the frequency of the Q is which would be white, is 0.2, just like we saw in the last example. And here's a picture of them, basically. So here are the frequencies at the beginning, 0.8 and 0.2. And here's the alleles in the population. And then they produce gametes. And each egg has an 80% chance of having a red allele and 80% chance of having a white allele. And same with the sperm. And so then the frequency of genotypes then can be calculated this way. Basically, this is an equation: p squared plus 2pq equal p squared plus 2pq plus q squared. That's basically a quadratic equation. And so, p squared then, well, p is 0.8. So p squared is 0.8 times 0.8, which is 0.64. And 2pq, so p is 0.8, right here. Q is 0.2, right? So we got 0.8 times 0.2, but then it's 2pq, so we have to multiply that by 2. And if you get out your calculator and multiply that out, 2 times 0.8 times 0.2, you're going to get 0.32, unless your calculator is broken. And then here, with the white alleles, Q squared, in other words, 0.2 times 0.2 equals 0.04. And so the frequency of genotypes then can be confirmed using a Punnett square. So basically then what this is saying is, if your red allele is at 0.8 proportion, and then the white allele, the proportion of the white allele is 0.2 in the population, then if everything is working right, and it's basically then 0.64 are going to be homozygous red, 0.32, the 0.32 proportion is going to be heterozygotes, and 0.04 proportion, in other words, if you convert it to percentages, 4%, 4% of the population would actually be homozygous white. And so if you go to Punnett square, set up a Punnett square, then 80% are going to be red, 20% are going to be white, and the same thing with the eggs. And so 0.8 times 0.8 is going to give you 0.64. 0.8 times 0.2 is going to give you 0.16. And then you add 0.16 to 0.16 to get the 0.32 proportion for the heterozygotes. And then, the same calculations for the white ones then. And so you get, the same, so with the Punnett square, then you get the same results as we got in our calculations on slide 40. So then, if P and Q represent the relative frequencies of only 2 possible alleles in a population at a particular locus, then P squared plus 2pq plus Q squared equals 1. Now, if there were 3 possible alleles, well then we'd have to have a different equation, P, Q, and R maybe. So where P squared, so this, we have this equation, and remember, P squared and Q squared represent the homozygous genotypes. and 2pq represents the frequency of the heterozygous genotype in the population. So this is the expected frequency of this genotype, RR. This 2pq is the expected frequency of R and W genotype, and Q squared then is expected frequency of W and WW, homozygous white genotype. So basically, here are the 5 conditions that have to be in place in order for any population to be in Hardy-Weinberg equilibrium, they call it. So there have to be no mutations going on, and that doesn't happen very frequently. There has to be random mating. Mating has to be absolutely random. There has to be no natural selection going on, which is not very common. They have to have an extremely large population size, so that things happen in totally randomly. There has to be no gene flow as well. So the consequences then, if so, the condition is no mutations, but if there are mutations, well then new mutations are going to come in and modify the gene pool. If mating is not random, then maybe random mixing of gametes does not occur and genotype frequencies might change. Now here, there should be no natural selection if they're in Hardy-Weinberg equilibrium, but if there is natural selection, well then probably allele frequencies are going to change. In order to be in Hardy-Weinberg equilibrium, they have to have a very large population size, and if the population size is not large, but rather it's small, well then the low frequencies can change over time, and we'll see that later on in the chapter. And here there's a condition of no gene flow, because if there is what they mean by gene flow is alleles that are moving in or out of the population. And if there's gene flow, if there's movement of alleles in or out of the population, that could change allele frequencies in that population. So now another thing is, natural populations can evolve at some loci while being in Hardy-Weinberg equilibrium and not evolving at other loci. So they don't have to be. A population doesn't have to be evolving at all of its loci all at once, and probably it's not. So the equation then can be tested, can be used then to test whether evolution is occurring in a population. And it's also used to determine the percentage of a population that's carrying a specific allele. We'll see that in part 2 of the lecture. So if you have any questions, though, we're going to end right now. We're going to end part 1 of the lecture right here and right now. But if you have any questions, though, about this, let me know. Send me an email or whatever. And so in probably 1 or 2 days, I will actually put up, I will record and put up lecture 2 then of chapter 23. So I will see you then. As always, like, comment, and subscribe.